Okay, so good evening everyone. Let's start today's live session. I'll share my screen and one of you can unmute and let me know if I'm visible and if my slides are visible and if my voice is audible. Right? So this is my screen. I hope my screen is visible and yeah, I hope my screen is visible. And my my screen is visible and I'm audible. Okay. Now, good evening, everyone. Today is the seventh live session of the series of live sessions where we basically discuss some old assignment questions of this particular course, post pathogen interaction, taken by course instructor Professor Himanshu Kumar from Aishi Bhopal. So my name is Aishi and I am a PMRF research scholar from the Department of Chemistry, IIT Bombay. So what we do in these sessions is that we discuss some old assignment questions and we discuss not only the answers to those questions but we also try to discuss the concepts behind those questions so that if similar type of other questions come, we can solve that easily, right? So that is the entire purpose of these live sessions. So moving on to the first question of this live session. Also we try to keep the sessions as interactive as possible. So if you have a doubt in any of such question, then you can unmute yourself and ask me. If I know the answer right now, then I'll answer you. If I don't, then you have to give me some time. I'll read about it and let you know by the next class. Okay. So that's how we will go in these sessions. Also very important. If you know the answer, so what the course this course is this these sessions are very interactive. So if you know the answer to the question, I'll see the question, then I'll stop and I'll let you answer the question. And then if you know the answer to the question, you can just let me know and then we'll move ahead and discuss accordingly. Okay. So moving on. The first question of today's session is which of the following are functions of the lymphatic system? There can be more than one correct answer. Okay, and the options are A. Reclaiming excess interstitial fluid to maintain fluid homeostasis. B. Facilitating antigen presentation in lymph nodes to activate immune responses. C. Producing red blood cells in response to low oxygen levels. D. Absorbing dietary fats from the intestines. E. Removing carbon dioxide from the bloodstream. F. Preventing tissue swelling through the drainage of excess fluid. G. Producing hormones to regulate metabolism. And 8. Synthesizing antibodies directly in the lymph nodes. Which one do you think are the correct answers? and you have to justify your answer as well. I'll wait for some time. Anybody? Anybody would like to try? Okay, so basically the options will be these. Why the first option is correct, reclaiming excess interstitial fluid to maintain fluid homeostasis, because the lymphatic vessels collect excess interstitial fluid that leaks out of blood capillaries and return it to the bloodstream, and this prevents fluid accumulation and helps maintain fluid balance in tissues. First of all, option B is also correct because facilitating antigen presentation in lymph nodes to activate immune responses is also right, because lymph nodes contain immune cells such as dendritic cells, macrophages, and lymphocytes, and antigens carried in lymph are presented to T cells and B cells, initiating adaptive immune responses. Right? Option C is not correct. Producing red blood cells in response to low oxygen levels is not correct, because red blood cells are produced in the bone marrow through erythropoiesis, regulated by erythropoietin from the kidneys, not the lymphatic system. Again, option D is correct, absorbing dietary fats from the intestines. Specialized lymphatic vessels called lacteals in the small intestine absorb dietary lipids and fat-soluble vitamins. These fats are transported as chylomicrons via lymph or chyle into the bloodstream. So that's correct. Option E is not correct. Removing carbon dioxide from the bloodstream is not correct, because carbon dioxide removal occurs in the lungs to the respiratory system, not the lymphatic system. Option F again is correct, preventing tissue swelling through the drainage of excess fluid, because by draining excess interstitial fluid, the lymphatic system prevents edema or tissue swelling. Option G and H are again incorrect, because producing hormones to regulate metabolism is not correct. Hormone production is a function of the endocrine system, not the lymphatic system. And option 8 is also incorrect, synthesizing antibodies directly in the lymph nodes. Antibodies are produced by the plasma cells, which are differentiated B cells. While this can occur within lymph nodes, the lymphatic system itself does not synthesize antibodies, rather immune cells do. So the final options, correct options will be these four. Okay, 1 second. Yeah, okay, sorry. So the final correct answer will be these four. So if you have any, okay. So like we were talking about the structure and the functions in the lymphatic system, right? So by the end of this PPT, you will be able to outline the structure of the lymphatic system, describe the functions of the lymphatic system, describe the structure of a lymph node, explain the functions of a lymph node, and state the location of the major lymph nodes. Okay, that is the purpose of this exercise. Now, the lymphatic system is part of the vascular system with three main functions. A. Returning tissue fluid to the bloodstream. B. Filtering fluid to prevent infection of the blood and tissues. C. Absorbing lipids from the small intestine via lacteals. Now, the lymphatic system is essential for preventing and reducing edema, that is, excess tissue fluid, maintaining blood viscosity, and vascular homeostasis. Now, what is the location of the lymphatic capillaries? So, if you see, this is the venule, the tissue cell. This is blood, and this is the interstitial fluid, and this is the lymph, right? Again, this is the blood capillaries, arterial, blood, lymph. This is the lymphatic capillary, right? Now, what is the structure of the lymphatic capillaries? So, lymph capillaries are blind-ended and have tiny flap-like non-return valves along their length. These are mini flap-like valve. Now, as tissue fluid around cells increases, pressure opens the flap-like valves and it enters the lymphatic system and is known as lymph. Okay, as you can see over here. Now, this opening is large enough to allow large molecules, bacteria, viruses, and other waste products to enter the system. Now, the fluid is named by location. If you can see, blood capillary, plasma is the tissue fluid, and lymph capillary will be lymph. Now, passage of lymph. So, once tissue fluid passes into the lymphatic system or lymph, it passes through a network of gradually increasing vessels in exactly the same manner as that of venous return. Resembling capillaries to vessels to trunks to veins. So, lymph returns to the bloodstream via two ducts into the subclavian veins, right lymphatic duct and the thoracic duct. Now, what is the movement of lymph through the system? Lymphatic circulation is not powered by a pump like the arteries, but works in a similar way to veins using following the means to move it through the lymphatic vessels. Option A, peristalsis of smooth muscle walls of the vessels. Option B, pumping from skeletal muscles. Option C, suction from the diaphragm. Option D, non-return valves. Option E, gravity. Okay. Now, movement of lymph through the system. Now, lymph contains many waste products, right, and pathogens that need to be removed before it re-enters the blood vascular system. And this is the role of lymph in the immune system. The lymph nodes are small bean-shaped structures in which these pathogens, old cells, and other waste products are neutralized and processed. The lymph nodes produce lymphocytes that fight infection by destroying pathogens such as bacteria. Now, what are the structure and function of lymph nodes? Functions: it can filter lymph, it can produce and store lymphocytes which fight infection. This is the direction of lymph flow in, and this is the direction of the lymph flow out. Now, what are the locations of the major lymph nodes? You can see axillary, cubital, inguinal, popliteal, auricular, occipital, cervical, submandibular. Now, associated lymphoid organs. Organs containing lymphoid tissue that support immune function. A. Spleen. B. Thymus. C. Tonsils. Now, organs containing lymphoid tissue that support immune function. Spleen. Spleen is located in the upper left abdomen. Stores lymphocytes to release as part of the immune response. It removes the worn out blood cells. Tonsils are up to 5 tonsils are located in the mouth. They can provide immunity to inhaled or ingested pathogens. Now, associated lymphoid organs, thymus, located against the trachea above the superior vena cava. They are prominent in infancy, providing immunity. They can aid lymphocyte white blood cells maturation. Programs T cells, aggressive white blood cells. Okay. So now can you outline the structures of the lymphatic system as we said in the beginning of the slide, and describe the functions of the lymphatic system, describe the structure of a lymph node, and explain the functions of a lymph node, state the location of the major lymph nodes, all of that. Okay. So the lymphatic system, which consists of vessels and organs, plays three vital roles in the body. We have discussed: protect the body against infection by activating defense mechanism that comprise of immune system, collect tissue fluids, solutes, hormones, and plasma proteins and return them to the circulatory system. They can also absorb fat from the small intestine into the lymphatic lacteals, if you can see over here. This video is not working. Now it can consist of lymph, which is a watery fluid that resembles plasma that contain fewer proteins and may contain fat together with cells. Then we have lymphocytes. Cellular components of lymph include T and B cells. Then we have lymphatic vessels, lymphoid tissues and organs. Now lymph node, important lymphocytes of the immune response are matured here. Spleen destroys RBCs and reservoir of blood. It is the largest lymph organ and it filters blood of bacteria and antigen filled cells. Now thymus gland produces hormone thymosin functions in programming lymphocytes T and B cells. T cells matured here like became immunocompetent. Now tonsils traps bacteria and other microbes in throat. Now there's Pear's patch which basically captures and destroys bacteria in the intestine, thereby preventing them from penetrating the intestinal wall. These are the Pear's patches. Now lymph, which is the excess tissue fluid carried by lymphatic vessels, is general definition. Now what are the properties of the lymphatic vessels? One-way system towards the heart, no pump, lymph moves towards the heart. So milking action of skeletal muscle and rhythmic contraction of smooth muscle in vessel walls. And what is the composition of lymph? So lymph is usually a clear colorless fluid similar to blood plasma but low in protein. Its composition varies from place to place. After a meal, for example, lymph draining from the small intestine takes on a milky appearance due to lipid content. Now, lymph may contain macrophages, viruses, bacteria, cellular debris, and even traveling cancer cells. Now, if we talk about lymph capillaries, walls overlap to form flap-like mini valves, and the fluid leaks into the lymph capillaries. Capillaries are anchored to connective tissue by filaments. Higher pressure on the inside closes mini valves. Now, what about the circulation in the lymph vessels? So, in the lymph nodes take the germ-filled lymph and filter the lymph before it is returned to the blood. Now, defense cells within lymph nodes. Macrophages engulf and destroy foreign substances. Lymphocytes provide immune response to antigens. Most are kidney-shaped, less than 1 inch long. Now, where are these lymph nodes? Spleen. So spleen, what does spleen do? It filters blood of bacteria, viruses and other debris. It destroys worn out blood vessels, like it then returns or recycles some of the breakdown products of RBCs to the liver. For example, iron, so that more RBCs can be made. And the unusual portion of worn out blood cells is excreted in bile. Now, what is another function? Stores platelets and acts as a blood reservoir. And lymphocytes are produced, RBC is also made in the fetus only. Now, edema is the excessive accumulation of fluids in tissue spaces. This can retard normal exchange of nutrients and metabolites. Anything that causes excess increased capillary pressure, such as decreased plasma protein, increased capillary permeability, or lymphatic blockage, can result in swelling and congestion of the extravascular compartment. As I said, the functions are draining excess tissue fluid. Not all fluid returns to the bloodstream and extra enters the lymphatic vessels. Now, immune response to eliminate pathogens from the body, innate and adaptive immune response. Major structures include lymphatic vessels, lymph nodes, spleen, and thymus. Lymphatic structures are lymphatic vessels. As I said, lymphatic tissues and organs like primary or central, bone marrow and thymus, and secondary or peripheral lymph nodes, spleen, and mucosa. The lymphatic vessels are very similar to veins, thin with valves. Unlike veins, they are a one-way street, originate as capillaries in the tissues, end at the veins. So, lymph is tissue fluid that has entered the lymphatic vessels with excess tissue fluid. Now, it transports proteins and other molecules to the bloodstream and moves foreign particles to lymph nodes. It is located now. Thymus is located behind the sternum, larger in children, and site of T cell maturation migrate, and they migrate from the bone marrow. Now, lymph nodes are located along lymphatic pathways, absent in the central nervous system are the lymph nodes. Their functions are that they filter and remove partially harmful particles before they enter bloodstream. Like there's an immune surveillance, monitoring of fluids by immune cells. Now, if you talk about spleen, it's located in the upper abdominal cavity, similar to the lymph node. The red pulp is majority of spleen, site of red blood cell disposal. Now, white pulp will be housing immunocytes, site of antigen presentation. Now, what do we know about mold or mucosa associated lymphatic tissue? So, lymphatic tissues in the mucosal membranes throughout the body contains T cells, B cells, and macrophages can encounter antigens passing through the mucous membranes. They are found in several portions in the body: lungs, digestive tract, nasopharynx, skin, breast, salivary glands. Now, what are the cells of the immune system? They are broken into two broad groups. Myeloid, mostly innate immune system, and lymphoid, adaptive immune system. Now, cells, they are basically mostly macrophages, engulf and kill invading microorganisms, can help induce inflammation and can activate other immune cells. For mast cells, they play a role in allergy responses, respond to parasitic worms, can induce inflammation. For granulocytes, which are basically WBCs, neutrophils, most numerous phagocytic, role in bacterial infection. For eosinophils and basophils, their defense against the parasites contribute to allergy response, can be more damaging than helpful. Dendritic cells basically can take up and degrade pathogens. Their main function is antigen presentation and activating cells of adaptive immunity. A bridge between the innate and the adaptive responses can be seen. Now, what are natural killer cells? They are not a part of the adaptive immune system but come from a lymphoid lineage, not antigen specific. They can recognize and kill some abnormal animals, tumor cells, and viruses, or to keep viruses at bay until adaptive response kicks in. Then there are the B lymphocytes. Antigen-specific receptors, B cell receptors, once activated, when activated, divides to form plasma cells, produce antibodies and secreted from the BCRs. Now, what are T lymphocytes or T cells? Categories are helper T cells, provide signals which initiate response from the other cells. Cytotoxic T cells kill cells which are infected with the virus or other pathogen. Regulatory T cells suppress immune activity and help control immune responses. Now, the immune system, cells and microbiochemicals responsible for the surveillance and destruction of the non-cell. Innate response, quick, non-specific, adaptive response, slower and specific. Now, what are the chemical signaling molecules? Proteins secreted which affect behavior of nervous cells, pro-inflammatory, regulatory or growth leaders, and then chemokines, proteins secreted with attract cells to an area, migrate due to a density gradient, cells have receptors that bind chemokines. Now, macrophages recognize pathogens through receptors, engulf and degrade pathogens. They can present antigens from the pathogen on the surface and secrete cytokines and chemokines. If we talk about inflammation, they can recruit proteins and cells to infected areas. Macrophages and neutrophils increases lymph flow. Brings microbes and antigen presenting cells to lymphatic tissue can recruit effectors of adaptive immunity later on. Now, complement. Pathogens can trigger activation of complement proteins. Activation begins a cascade which can result in destruction of the pathogen, and it can also promote inflammation. Now, what are PAMs? Pathogen associated molecular patterns. Now, cells like macrophages, neutrophils, and dendritic cells have receptors that recognize simple molecules and regular molecular patterns. Pattern recognition centers or PRR. So, RAMs are made on microorganisms but not body cells. PAMs are made on microorganisms and not on body cells. Now, innate response initiates adaptive response. So, macrophages engulf pathogens and present antigens to lymphatic, dendritic cells triggered to activate T cells. Adjuvants in bacterial extracts is in vaccines initiate an immune response, which in turn activate the adaptive response that leads to successful vaccination to the purified protein. Now, initiated by APCs or antigen presenting system factors, antigen presenting cells, primarily dendritic cells but also macrophages, engulf pathogens, degrade, then move to the antigen to surface of the wall. Dendritic cells also have co-stimulatory molecules, stimulate its T cells to proliferate and differentiate. B cells usually also wear a helper T cell. Require a helper T cell. So it occurs in secondary lymphoid tissue or organs. Now, about proliferation, once activated, naive lymphocytes become lymphoblasts, can divide to produce clones, which is known as the clonal expansion. Can divide 2 to 4 times per 24 hours for about 3 to 5 days, producing about 1000 cells specific to the target antigen. These cells become effector cells. B cells are plasma cells. T cells heterotoxic or helper T cells. So most cells generated by clonal expansion die. Some T and B cells are left behind. These are memory cells. They can be reactivated quickly when the same antigen is encountered again. That provides long-lasting immunity. Now, what are the mechanisms of adaptive immunity? Cell-mediated immune response. T cell responsible for destruction of intracellular invaders. Humoral immunity is antibodies pathogens in the blood and other extracellular fluid. Other mechanisms are similar to those of the innate community. Macrophages, neutrophils complement. Then we have the T cell mediated immunity. Like CD4 is called the helper T cell, CD8 is called the cytotoxic T cell. Now, recognize peptides and MHC molecules, which are the major histocompatibility complex. The complex displays on surface of cells, trap peptide leading production and transported to surface. MHC class 1 protein synthesis in cholesterol in cytosol, can display viral proteins recognized by cytotoxic T cells. MHC class 2 is expressed by ABCs, antigen presenting cells that we talked about in the beginning, derived from proteins taken in through phagocytosis and endocytosis, can activate CD4 plus. Now, stimulated effector cells secreted Effector molecules, mainly cytokines, recruit other effectors to target area. CD8 cytotoxic T cells directly kill infected cells, recognize antigen. CD4 helper T cells can become different types of T cells. Now, Th1 help control bacteria in vesicles or macrophages of macro stimulate them to increase intercellular destruction of bacteria. Promote before the mucous services parasitic infections. So promote responses with lots of neutrophils, extracellular bacteria and fungi. The syndrome helper in lymphoid follicles and B cell activation. Now we have talked about the antibody structure. I'll not go into too much detail about the antibody structure. Not about the immunity section also because you've already read too much about it. Vaccines are also done. Lymphatic collecting vessels can collect lymph from the lymph capillaries, carries lymph to and away from the lymph node, and returns fluid to the heart through two ducts, right lymphatic duct. That drains lymph from right arm and right side of the head and thorax, thoracic duct, receives lymph from the rest of the body. We see this image also. So, what are the common features? Predominance of regular inactive tissues and lymphocytes. So, spleen filters blood of bacteria and viruses located on the left side of the alveolar cavity. Destroys worn out red blood cells. Acts as a blood reservoir. Produces ampullite lymphocytes. Sorry. And thymus basically functions peaks at youth, fall low and throat over heart. Producer hormones that program lymphocytes. So tonsils, small masses of lymphatic tissue around pharynx, trap and removes bacteria or pathogens entering that so inefficient can become congested. Tonsillitis. Pairs patches like pairs tonsils between small intestine of the captures. It captures and destroys bacteria. So coming back to the question that we started with, these are the four accepted or right answers. Anybody has any doubt? Anybody any doubt in this question? If not, then we'll move on to the next question for today. Any doubt? Anybody? If no answer, then no queries or no answer, we'll move on to the next question for today. Okay, there's no doubt in this question. We'll move on to the next question for today. Okay, sorry, once a minute. Sorry, once again, there was network issue. I will share my screen again. Yeah, this was a question. This was the answer. Okay, so moving on to the next question for today. Which of the following statements best describes the role of M cells in the immune system? A. M cells produce antibodies to neutralize pathogens at mucosal surfaces. B. M cells secrete cytokines that activate nearby lymphocytes in the gut associated lymphoid tissue. C. M cells facilitate the transcytosis of antigens and pathogens across the epithelial barrier to immune cells in pairs patches. D. M cells function primarily to phagocytose and destroy pathogens at the mucosal surfaces. E. M cells are specialized in producing IgA or immunoglobulin A antibodies in response to mucosal infections. I'll wait for a few minutes. If you want, you can answer me. Anybody? So the correct answer will be M cells facilitate the transcytosis of antigens and pathogens across the epithelial barrier to immune cells in the pairs patches. So what happens is that M cells, which are microfold cells, are specialized epithelial cells found in the follicle associated epithelium of pairs patches in the intestine, right? So their primary function is antigen sampling. So they capture antigens, microbes, and particulate matter from the intestinal lumen and transport them across the epithelial barrier via transcytosis. Now, these antigens are delivered to dendritic cells, macrophages, and lymphocytes located in the underlying gut associated lymphoid tissue or GALT, which then initiates the immune responses. Other options are incorrect. Option A is incorrect because antibodies are produced by plasma cells, activated B cells, not M cells. Option B is incorrect because cytokines secret cytokines to activate lymphocytes. Cytokines are mainly produced by immune cells such as T cells, macrophages, and dendritic cells, not M cells. Option D is not correct because that is phagocytosis destroy pathogens. Not correct because macrophages and neutrophils perform phagocytosis. M cells mainly transport, not destroy pathogens. Option E is also not correct because producing IgA antibodies, IgA is produced by plasma cells in mucosal tissues, not by M cells. So the final answer can be C only. Now, all cells in the immune system are derived from stem cells and either myeloid or lymphoid precursors under the control of cytokines. Now, neutrophils, mast cells, basophils, and eosinophils are important granulocytic cells involved in phagocytosis or release of innate immune inflammatory mediators. Now, neutrophils normally found in circulation migrate out of blood vessels to sites of infection in effort to phagocytose invading bacteria. Now, mast cells release histamine, cytokines, and other inflammatory mediators when IgE bound to the surface of the cell is cross-linked by an allergen. Now, monocytes are quiescent precursors to macrophages and are found in all tissues in the body. On activation by invading pathogens, cytokines, etc., monocytes differentiate into macrophages and angry monocyte that can phagocytose basically pathogens and present antigen to T cells to engage the adaptive immune response. T and B cells are the main cell types of the adaptive immune system. B cells express membrane-bound antibody IgM on their surface to recognize intact unprocessed antigen. T cells express a T cell receptor that recognizes processed antigen bound to the MHC molecules. Now, what are the primary lymphoid organs? Thymus, encapsulated organ where T cells mature and are selected based in part on their ability to not recognize the host. Then we have bone marrow, site of remaining hematopoiesis. B cells mature here mostly in skull, ribs, sternum, and pelvis. Now, what are the lymphatic vessels? They drain the entire body to a regional lymph nodes. Is a secondary circulatory system moving antigen to lymph nodes where B and T cells can respond. Now, if we talk about lymph nodes, encapsulated organ found throughout the body. Again, from the periphery is moved here through lymphatics by antigen presenting cells. Lymphoid follicles, collection of T and B cells in the node on encountering the specific antigen will proliferate, generating a germinal center that leads to the formation of fully mature effector T and B cells. If we then talk about spleen, encapsulated organ found under the liver, highly vascular organ that filters blood, is responsible for eliminating blood-borne pathogens. Now, RBCs carrying immune complexes are processed here to remove antigen, and it also has follicles that on encountering the specific antigen will proliferate, generating a terminal center that leads to the formation of fully mature effector T and B cells. Now, if we talk about Peyer's patches, lymphoid tissue in the small intestine composed of T and B cells and antigen presenting cells not encapsulated. It's part of the mucosal associated lymphoid tissue or mold. Now, pathogens move through M cells where they encounter elections of lymphocytes that can generate a germinal center on encountering this specific antigen. Now, responses here usually generate IgA producing B cells. Okay. So, the large, as I said, if you see about all the The large intestine of the colon has a large resident population of microbiota consisting of at least 10 to the power 12 organisms per gram of luminal contents. And these organisms together with the antigenic load provided by the diet and the constant threat of potential pathogens means the intestinal immune system encounters more antigen than any other part of the body. Now, if you talk about the balance, so as many pathogens enter the body via intestinal mucosa, it is vital that the gut associated lymphoid tissues can provide effective immune responses when necessary. However, inappropriate responses against innocuous food and commensal antigens lead to inflammatory disorders such as celiac disease and inflammatory bowel disease. Now, what do we know about the gut associated lymphoid tissue? The lymphoid elements of the gut comprise organized lymphoid tissues such as the pairs, patches, PP, and the mesenteric lymph nodes. MLN. The effector sites of the intestine are the mucosal epithelium and underlying lamina propria. Here there are many different immune cells including activated T cells, plasma cells, mast cells, dendritic cells and macrophages even under normal conditions. So that this does not result in overt tissue pathology reflects the fact that the effector cells present are actively held in check by potent regulatory mechanisms. Now, if we talk about immune protection in the gut, although composed of only a single cell layer, the intestinal epithelium forms a barrier against penetration of microbes. The defects in barrier function contribute to the development and perpetuation of inflammation in inflammatory bowel disease. So, epithelial cells of the small intestine are coated in a glycocalyx of mucins and other glycoproteins that can interact with and trap bacteria in the mucus. In addition, antimicrobial peptides such as defensins are secreted by Paneth cells located at the bottom of the intestinal crypts. Epithelial cells also act as microbial sensors by secreting factors such as IL-8, MCP1, RANTES, TNF-alpha and IL-6 in response to bacterial entry. So this results in the recruitment of neutrophils, eosinophils, monocytes, phagocytic macrophages and T cells and so enhances the induction of protective immunity. Although many of these cells are also present in the normal intestine under physiological conditions, they are conditioned by factors in the local environment to be non-inflammatory. So the normal and the inflamed intestine contains many specific immune cells including IgA secreting plasma cells, CD4 plus and CD8 plus T cells, regulatory T cells and gamma delta T cells. Okay, so as we said, commensal bacteria and pathogens share many factors which can be detected by pathogen recognition receptors, such as toll-like receptors. So how do commensals fail to trigger inflammatory responses? So modulation of innate activating receptors such as CD14 and CD89 on gut macrophages, high levels of immunomodulatory factors IL-10, TGF-beta, TSLP, retinoic acid which can condition local cells, reduced function of TLRs in intestinal DC. Commensals are also non-invasive, so whereas pathogens penetrate the epithelium and trigger inflammatory responses both locally and more widely, commensal bacteria only penetrate the epithelium after uptake by local dendritic cell and are then transported to the draining MLN where their progress is halted. So this results in the production of secretory IgA in the gut which limits commensal numbers and regulatory T cells which dampen inflammatory responses. Local non-inflammatory macrophages also ingest and kill the rare commensals which enter. Okay. So we started with different topics. So let's go back to the basics once more. Hematopoiesis. So all blood cells arise from a type of cells called the hematopoietic stem cell. It's remarkable that every functionally specialized mature blood cell arises from an HSC. Now, stem cells are cells that can differentiate into other cell types. They are self-renewing, maintaining their population level by cell division. Now, early in hematopoiesis, the multipotent stem cell differentiates along one of two pathways, giving rise to either a lymphoid progenitor cell or a myeloid progenitor cell. And progenitor cells have lost their capacity for self-renewal and are committed to a particular cell lineage. Now, what are lymphoid progenitor cells? They can give rise to B, T, and NK cells. Myeloid progenitor cells generate progenitors of the red blood cells, erythrocytes. Many of the various white blood cells, neutrophils, eosinophils, basophils, monocytes, mast cells, dendritic cells, and platelet generating cells called megakaryocytes. Hematopoiesis is regulated at the genetic level. Example of these is GATA2 gene. Okay, if you can see, these are the visualizing basically concepts. Whatever we have been discussing, hematopoietic stem cell which is self-renewing, and these are the two lineages, either myeloid progenitor or lymphoid progenitor, and so on and so forth. Lymphocytes are the central cells of the immune system responsible for adaptive immunity and the immunologic attributes of diversity, specificity, memory, and self-non-self recognition. Other types of white blood cells play important roles in engulfing and destroying microorganisms, presenting antigens and secreting cytokines. So lymphocytes constitute 20%, 40% of the body's white blood cells and 19% of the cells in the lymph. These lymphocytes continually circulate in the blood and lymph and are capable of migrating into the tissue spaces and lymphoid organs, thereby integrating the immune system to a high degree. Now, the lymphocytes can be broadly subdivided into three populations: B cells, T cells, and natural killer cells on the basis of function and cell membrane components. Natural killer cells are large granular lymphocytes that do not express a set of surface markers typical of B or T cells. Now, if you talk about B lymphocytes or B cells, it's derived its better designation from its site of maturation in the bursa of Fabricius in bird and in bone marrow, where it is the major site of maturation in a number of mammalian species including humans and mice. Now, mature B cells display membrane-bound immunoglobulin molecules which serve as receptor of Ag or BCR. Now, the binding of Ag to the AB causes the cell to divide rapidly. Its progeny differentiates into effector cells called plasma cells, which produce the antibody in a form that can be secreted and have little or no membrane-bound antibody. They are the end-stage cells and do not divide. Or there are memory B cells which have a longer lifespan than naive cells and they express the same membrane bound antibody as their parent B cell receptor. Now, again, T cell, it derives its literal designation from the site of maturation in the thymus. During its maturation within the thymus, the T cell comes to express on its membrane a unique antigen binding molecule called the T cell receptor. TCR recognizes antigen that is bound to cell membrane proteins called major histocompatibility complex MHC. There are two well-defined subpopulations of T cells, helper T cells and cytotoxic T cells. Recently, a third T cell population, regulatory T cells have come up, which T helper cells characterized by the presence of CD4 marker membrane glycoprotein on their surface, whereas T cytotoxic express CD8, and the ratio of Th to Tc is about 2:1 in normal human peripheral blood, but it may be significantly altered by immunodeficiency diseases, autoimmune disorders, and other diseases. Now, if we talk about the natural killer cells, basically they are lymphocytes that display cytotoxic activity against a wide range of tumor cells in the absence of any previous immunization with the tumor. So NK cells were subsequently shown to play an important role in the host defense both against tumor cells and against cells infected with some, though not all, viruses. Now, these cells, which constitute 5 to 10% of lymphocytes in human peripheral blood, do not express the membrane molecules and receptors that distinguish T and B cell lineages. They can recognize potential target cells in two different ways. Number one, a reduction in the display of class 1 MHC molecules. NK cells express the non-TCR related receptor called killer cell inhibitory receptor which bind to MHC class 1 antigen. Number two, the immune system has made an anti-tumor or antiviral antibodies that are bound to their surface because NK cells express CD16, a membrane receptor for their Fc region of the IgG molecule. They can attach to these antibodies and subsequently destroy the targeted cells. This is an example of a process known as antibody-dependent cell-mediated cytotoxicity. Okay. Now, what are mononuclear phagocytes? The mononuclear phagocytic system consists of monocytes circulating in the blood and macrophages in the tissue. So, phagocytosis of particular antigen serves as an initial activating stimulus. Macrophage activity can be further enhanced by cytokines secreted by activated Th cells, by mediators of the inflammatory response, and by components of bacterial cell walls. Now, one of the most potent activators of macrophages is interferon gamma secreted by activated Th cells. Activated macrophages also express higher levels of class 2 MHC molecules, allowing them to function more effectively as antigen presenting cells. Thus, macrophages and Th cells facilitate each other's activation during the immune response. Activated macrophages secrete a collection of cytokines such as interleukin 1, TNF alpha, and interleukin 6 that promote inflammatory response. So, complement proteins, hydrolytic enzymes contained within the lysosomes of macrophages, and TNF alpha that can kill a variety of cells. Now, macrophages are dispersed throughout the body. Some take up residence in particular tissues, becoming fixed macrophages, whereas others remain motile and are called free or wandering macrophages. Free macrophages travel by amoeboid movement throughout the tissues. Macrophage-like cells serve different functions in different tissues and are named according to their tissue location. Alveolar macrophages in the lung, histiocytes in connective tissues, Kupffer cells in the liver, mesangial cells in the kidney, microglial cells in the brain, and osteoclasts in bone. Now, if we talk about the granulocytic cells, the granulocytes are classified as neutrophils, eosinophils, and basophils on the basis of cellular morphology and cytoplasmic staining characteristics. Okay, we have talked about neutrophils. They are produced by hematopoiesis in the bone marrow. In response to many types of infections, the bone marrow releases more than the usual number of neutrophils, and these cells generally are the first to arrive at a site of inflammation. And the resulting transient increase in the number of circulating neutrophils, called leukocytosis, is used medically as an indication of infection. Now, movement of circulating neutrophils into tissues called extravasation takes several steps. Number 1, the cell first adheres to the vascular endothelium. Number 2, then penetrates the gap between adjacent endothelial cells lining the vessel wall. Number 3, and finally penetrates the vascular basement membrane, moving out into the tissue spaces. A number of substances generated in an inflammatory reaction serve as chemotactic factors that promote accumulation of neutrophils at an inflammatory site. For example, complement components. Now, like macrophages, neutrophils are active phagocytic cell. Phagocytosis by neutrophils is similar to that described for macrophages, except that the lytic enzymes and the bactericidal substances in neutrophils are contained within primary and secondary granules. Neutrophils are in fact much more likely than macrophages to kill ingested macroorganisms. Neutrophils exhibit a larger respiratory burst than macrophages and consequently are able to generate more reactive oxygen intermediates and reactive nitrogen intermediates. Eosinophils, like neutrophils, are motile phagocytic cells that can migrate from the blood into the tissue spaces. The phagocytic role is significantly less important than that of the neutrophils, and it is thought that they play a role in the defense against parasitic organisms. Now, basophils are non-phagocytic granulocytes that function by releasing pharmacologically active substances from the cytoplasmic granules. These substances play a major role in certain allergic responses. A mast cell precursors, which are formed in the bone marrow by hematopoiesis, are released into the blood as undifferentiated cells. They do not differentiate until they leave the blood and enter the tissues. It plays an important role in the development of allergies. The new dendritic cell acquired its name because it is covered with long membrane extensions. There are many types of dendritic cells, although most mature dendritic cells have the same major function, the presentation of antigen to Th cells. Four types of dendritic cells are known: Langerhans cells, interstitial dendritic cells, myeloid cells, lymphoid dendritic cells. Each arises from hematopoietic stem cells via different pathways and in different locations. They all constitutively express high levels of both class 2 MHC molecules and members of the co-stimulatory B7 family. For this reason, they are more potent antigen-presenting cells than macrophages and B cells, both of which need to be activated before they can function as antigen-presenting cells. Another type of dendritic cell, the follicular dendritic cell, does not arise in bone marrow and has a different function from the antigen-presenting dendritic cells. Follicular dendritic cells do not express class 2 MHC molecules and therefore do not function as APCs. For Th cell activation, it expresses high levels of membrane receptors for antibody, which allows the binding of Ag antigen-antibody complexes. The interaction of B cells with this bound antigen can have important effects on B cell responses. So, we have already talked about the organs of the immune system. This can be distinguished by fraction as the primary and secondary lymphoid organs. The thymus and bone marrow are the primary or central lymphoid organs, where maturation of lymphocytes takes place. The lymph nodes, spleen, and various mucosal associated lymphoid tissues or MALT, such as gut associated lymphoid tissue or GALT, are secondary peripheral lymphoid organs which trap antigen and provide sites for mature lymphocytes to interact with that antigen. In addition, tertiary lymphoid tissues, which normally contain fewer lymphoid cells than secondary lymphoid organs, can impart lymphoid cells during an inflammatory response. Most prominent of these are cutaneous associated lymphoid tissues. Now, once mature lymphocytes have been generated in the primary lymphoid organs, they circulate in the blood and lymphatic system, a network of vessels that collect fluid that has escaped into the tissues from capillaries to circulatory system and ultimately return into the blood. Okay. We'll not go into too much detail about this. We have already discussed about this. I upload the slides so you can check out. You'll be able to check out the all these slides. I'm not going into too much detail. We have discussed about all of these in one or previous classes, so I'll not repeat the same thing. Okay, coming back to the question that we started with. Which of the following statements best describes the role of M cells in the immune system? M cells facilitate the transcytosis of antigens and pathogens across the epithelial barrier to immune cells in the periaspais patches. So, anybody has any question related to this? If not, then we'll move on to the next question for today. I'll wait for a few minutes. If you have any question, you can ask. Okay. See, if there are no questions, we move on to the next and the last question for today. So, this question is, if a knockout gene affects embryo development in the animal model and results in lethality, which strategy for gene knockout could be useful? A. Knock in, B. Conventional knockout, C. Depletion of hematopoietic stem cells, D. Conditional knockout, and E. Not possible to make knockout for such condition. You have to answer in detail. I'll wait for some time. One second. Okay, so the answer will be conditional knockout. Okay, so if a gene knockout causes embryonic lethality, it means that the gene is essential for early development. In such cases, removing the gene in all the cells from the beginning, like germline knockout, prevents the embryo from surviving. So, researchers cannot study the gene's function in adult tissues. But why a conditional knockout works? A conditional knockout allows researchers to delete the gene only in specific tissues or at specific times rather than throughout the whole organism. And this is typically achieved using systems such as the Kree-LoxP recombination system. The target gene is flanked by LoxP sites, floxed gene. Another mouse line expresses Cree recombinase under a tissue-specific or inducible promoter. So when Cree is expressed, CRE, it excises the gene only in those specific cells or at a chosen developmental stage. This allows the embryo to develop normally, the gene to be removed later or in a particular tissue, enabling functional studies. For example, a gene essential during embryogenesis can be knocked out only in adult liver cells, neurons, or immune cells. Right? So, the key concept is that when a gene knockout causes embryonic lethality, researchers use conditional gene knockout systems to study gene function in specific tissues or later developmental stages without killing the embryo. So, what are knockout mice? A knockout mouse is a mouse in which a specific gene has been inactivated or knocked out by replacing it or disrupting it with an artificial piece of DNA. The loss of gene activity often causes changes in a mouse's phenotype and thus provides valuable information on the function of the gene. Now, who are the researchers who actually made this concept come to life? The researchers who developed the technology for the creation of knockout mice won the Nobel Prize in the year 2007. So the Nobel Prize in Physiology or Medicine 2007 was awarded jointly to Mario R. Capecchi, Sir Martin J. Evans, and Oliver Smithies for their discoveries of principles for introducing specific gene modifications in mice by the use of embryonic stem cells. So, the ability to delete or mutate any gene of interest in mice has transformed the landscape of mammalian biology research. Now, the cultivation of embryonic stem cells, Martin Evans, gene targeting, Oliver Smithies, and gene knockout is Mario Capecchi. Okay, these are some of the papers, gene correction by Oliver Smithies and all. Gene knockout by Mario Capecchi. These papers came out in Nature and all. You guys can go through these papers. Now, what is the importance of this technique, right? So, knocking out the activity of a gene provides valuable information about the functions of this gene, right? Since many genes are in common between humans and mice, knockout mice gives researchers information about the function of that gene in humans. Now, knockout mice have been useful and in studying and modeling different types of cancer, obesity, heart disease, diabetes, arthritis, anxiety, aging, and Parkinson disease. Now, knockout mice also offer a biological context in which drugs and other therapies can be developed and tested. Now, transgenic versus knockout mice. A transgenic mouse typically expresses one or more copies of a gene, cDNA, that is integrated into its genome in a random fashion. A knockout mouse is a mouse in which both alleles of a gene are deleted in a targeted fashion by homologous recombination. Now, while traditional transgenic mice are generated to express a protein, much information can be learned from the elimination of a gene or the deletion of a functional domain of the protein. Now, creation of transgenic mice involves injection of DNA into fertilized eggs and looking for adult animals that produce a protein of interest. In transgenic mice, the transgene gets inserted into the genome randomly. Thus, transgenic mice were limited to gain of function mutations, wherein two copies of the normal gene are joined by additional copies which would overproduce the normal or the mutant protein. So, using transgenic mice, one can infer a gene's function by observing what happens when there was too much of the protein or when a mutant copy interfered with the normal one. Now, the gene knockout technology allowed researchers to delete specific genes In this case, the transgene, which is usually a mutant or deleted version of the normal gene, is introduced into embryonic mouse cells in culture and are then allowed to recombine with its sister DNA in the genome. The recombination cuts out the resident DNA and inserts the mutant DNA, which can then form a damaged protein or no protein at all. Now, the embryonic stem cells then grow into a whole mouse with a specific mutation exactly where it is supposed to be in the genome. Thus, gene knockout technology allowed researchers to study loss of function mutations, wherein one can infer a gene's function by observing what happens when the gene is absent or when mutant copy of the gene is expressed instead of the normal one. The first human disease model created with this technology was cystic fibrosis from the labs of both Smithies and Evans. So, if you see a laboratory mouse in which a gene affecting hair growth has been knocked out, is shown next to a normal lab mouse, right? A knockout mouse left, that is a model of obesity compared with the normal mouse. So, knocking out expression of NHLH2, a basic helix to helix transcription factor in the mice, results in adult onset diabetes. Okay, so this knockout mice at 8 weeks of age are similar to normal mice in weight and body structure, but older knockout animals have an increase in abdominal fat and body weight. You see, more than twice the muscle mass of wild type mouse. This is FG knockout mouse is long angora like hair. These are so knocking out P53 gene. So it was well known that P53 is a tumor suppressor gene and is deleted or mutated in half of the human cancers. It was therefore of interest to see what happens if you delete P53 gene in mice. So P53 knockout mice developed normally but developed a variety of cancers including lymphomas when they grew old. Thus, the knockout model provided the ultimate proof that P53 is indeed a tumor suppressor. One important result the gene knockout models revealed is that cells need more time, more than one mutant gene to become cancerous. When you knock out P53, it takes many months for cancer to arise. The cells need more genetic alterations than just one. Since mice engineered to express a mutant RB gene indeed developed tumors, but not the ones researchers were expecting. Instead of eye tumors, the animals suffered from pituitary and thyroid gland cancers. It was later found that the second gene protected the eye cells from cancer, and both mutations were required for the tumors to form. In another study, when the gene encoding telomerase, the protein responsible for keeping chromosome tips long and healthy, was knocked out in mice. In such mice, cancers arose when chromosome tips shorten over the course of a lifetime of cell division. The failing tips cause chromosomes to break and recombine incorrectly until finally cells reach the large number of mutations needed for transition to malignancy. The most important factor that drives cancer is age. The knockout mice models also contribute to understanding the function of genes in development. Very often, oncogenes were found to be critical in normal development. A generation of knockout mouse by homologous recombination. What is the methodology? Now, basically creating a knockout construct, you need that first. Introducing the knockout construct into mouse embryonic stem cells in culture, screen ES embryonic stem cells and select those whose DNA includes a new gene. Implant selected gene cells into normal mouse embryos, making chimeras. Implant chimeric embryos in pseudo-pregnant females. and females then can give birth to chimeric offspring which are subsequently bred to verify transmission of the new gene, producing a mutant mouse line. So gene knockout is basically accomplished by homologous recombination involving a double crossover event. So if someone tells what do you mean by gene knockout is accomplished by homologous recombination involving a double crossover event. So if you want me to explain in detail regarding this statement, what this means is that when we say a knockout is accomplished by homologous recombination involving a double crossover event, we mean that a target gene in the genome is replaced or disrupted by an engineered DNA construct through recombination of two matching DNA regions. Now let's break it down clearly. One will be homologous recombination. Homologous recombination, as you can see here, is a natural DNA repair process where two DNA molecules with similar or identical sequence exchange segments. In gene knockout experiments, scientists design a targeting vector like DNA construct, and this vector contains DNA sequences identical or homologous to regions flanking the gene of interest. Now, these homologous regions guide the construct to the correct location in the genome. Now then, the structure of the targeting vector. A typical knockout construct contains left homologous arm, sequence identical to DNA upstream of the target gene, then selectable marker like neomycin resistance gene replaces or disrupts the target gene, then right homologous arm, sequence identical to DNA downstream of the target gene. So the construct looks like left homology arm, selection marker, disrupting gene, right homology arm. Then there is the double crossover event. During homologous recombination, two crossover events occur. First crossover between the left homologous arm and the corresponding genomic region. Second crossover between the right homologous arm and the downstream genomic region. Because recombination occurs at 2 points, the DNA segment between them is replaced. So original genome, upstream DNA, target gene, downstream DNA becomes after double crossover. Upstream DNA selected marker, disrupted gene, downstream DNA. Thus the target gene is removed or disrupted, producing a knockout. Now why is it called double crossover? It is called double crossover because recombination happens at two homologous regions, two crossing points, one on the left side, one on the right side. And this ensures a precise replacement of the gene rather than random insertion. And why this method is important? Because this approach follows precise targeting of specific genes, creation of knockout mice or cell lines, study of gene function by observing the phenotype when the gene is absent. So this classical method was widely used in mouse embryonic stem cells before CRISPR-based gene editing became common. Okay, we will not go into too much detail. So basically, positive selection markers are used to enrich for recombination events. So expression cassettes encoding antibiotic resistance genes for neomycin. And negative selection markers used to enrich for homologous recombination events over random insertions. So like use of herpes simplex virus thymidine kinase gene coupled with ganciclovir treatment. So ganciclovir is a synthetic nucleoside analog of deoxyguanosine which is phosphorylated by HSV-TK and the metabolite inhibits DNA replication when incorporated into DNA by termination of DNA elongation. Now, what are the drawbacks of knockout mice? Many genes that participate in interesting genetic pathways are essential for either mouse development, viability, or fertility. Therefore, a traditional knockout of the gene can never lead to establishment of a knockout mouse strain for analysis. About 15% of gene knockouts are developmentally lethal and therefore cannot grow into adult mice. Thus, it becomes difficult to determine the gene function in adults. To overcome these drawbacks, the conditional knockout approach was developed, which allowed researchers to delete the gene of interest in a time and space dependent manner using site specific recombinases. Using these recombinases, it is possible to knock out the expression of a gene in a specific mouse tissue or at a specific stage of development or in response to an inducer. That becomes the Krelok system for temporal control of the transgenic gene expression. So, recombinase is a site-specific integrase isolated from the P1 bacteriophage. It catalyzes the recombination between two of its consensus DNA recognition sites, known as the LOXP sites, which are 34 base pairs in length. They consist of two 13 base pairs palindromic sequences that flank a central sequence of 8 base pairs, which determines the directionality of the LOXP site. Now, two Loxp sites are most often placed in a transorientation on either side of an essential functional part of a gene. So, the recombination removes the functionality and knocks out the gene. Now, Loxp sites can also be placed in a cis orientation to invert the intervening sequence. Loxp sites placed on different chromosomes can be used to generate targeted translocations, though this recombination event occurs at a relatively low frequency compared to the highly efficient intragene recombination. Now, spatial and temporal knockout is achieved by the choice of promoter used to drive the CRE gene expression. Several mouse lines, each expressing CRE from a promoter that is either tissue specific, cell specific, developmentally specific, or responsive to an exogenous event like tetracycline, are now available. The several promoter specific mouse models can be generated. In contrast to knockout, in which a gene or a part of the gene is deleted, knock-in is the replacement of a gene by a mutant version of the same gene using homologous recombination. So, knock-in is very useful when establishing a disease model of a specific disease-related mutation in the human gene. More than 10,000 different genes in mice, approximately half of the genes in the mammalian genome, have been studied with gene targeting. Roadmap to disrupt each of the 20,000 genes in the mouse genome before 2010. Okay, I will not go into too much detail of this. I will share the slides with you. Coming back to the question that we started with, if a knockout gene affects embryo development in the animal model and results in a lethality, which strategy will be used for gene knockout could be useful, that will be conditional knockout. I'll wait for a few minutes. If you have any questions, you can ask me. Yes, can you type in the message if you have any question? And if you don't have any question, then we can stop the session here today. And we'll meet next Monday, same time. Thank you. Hello, I cannot hear you. Can you type it? Hello.
Yes, ma'am.
Your voice is cracking. Can
Yes,
you
ma'am.
type your question?
No, ma'am, I do not have any question. I am telling the answer.
Oh,
What I
acha,
am
it was already
using.
discussed. Yeah, the answer will be conditional knockout. Yes. I already
Yes.
discussed it and I have the recording as well. So I'll upload the
Okay, ma'am.
recording and I'll also share the slides.
Okay, ma'am.
Okay. Thank you. So we'll stop the session here. Next Monday we'll meet. I'll upload the sessions and the slides and the recording. Thank you.
Thank you, ma'am.